Today I am trying to take up a detailed study of homeopathic polycrest remedy sepia from an MIT perspective. Study of any drug substance from MIT perspective of scientific homeopathy has to be done by exploring the molecular constituents of the given drug substance, exploring their roles in the biochemistry of the living organism in molecular forms, and correlating them with the symptoms given in their homeopathic materia medica as far as possible. Such a study should also explain how the molecular imprints of those drug molecules contained in their potentized forms work as therapeutic agents by acting as artificial binding pockets for diverse types of pathogenic molecules having conformational affinity. Obviously it will be a tough task, as currently we lacking sufficient data regarding the molecular constituents as well as biological targets of most of the complex drug substances used in homeopathy. Moreover, we know very little about the biomolecular mechanisms that underlie the multitudes of objective and subjective symptoms collected by drug proving and listed in the homeopathic materia medica. As such, only a very generalized and limited explanation demonstrating the MIT approach could be expected here. All of us know, homeopathic drug sepia is prepared from ink of cuttlefish. Cuttlefish are marine animals of the order Cepida. They belong to the class Cephalopoda, which also includes squid, octopuses and nautiluses. Despite their name, cuttlefish are not fish but mollusks. Sepia ink is a dark pigment released into water by most species of cephalopod, usually as an escape mechanism. Its dark color is caused by its main constituent, melanin. It contains a number of other chemicals also in a variety of concentrations, such as tyrosinase, dopamine and L-dopa, and small amounts of amino acids, including taurine, aspartic acid, glutamic acid, alanine and lysine. Sepia ink also contains large amounts of aquatic minerals such as iodine, sodium, fluorine, iodine etc. absorbed from seawater in which they live. That means, sepia is not a single drug as we are made to believe. It is a highly complex compound drug. During drug proving, all these different chemical constituents of sepia act in their individual capacities upon different biological targets, produce molecular errors that are expressed through various groups of subjective and objective symptoms. When potentized, these different chemical molecules undergo molecular imprinting as individual molecules. As such, potentized sepia will be a combination of diverse types of molecular imprints that represent different types of constituent chemical molecules. When used as therapeutic agent, these individual molecular imprints bind to specific pathogenic molecules having complementary conformation. Tyrosinase is a rate-limiting enzyme for controlling the production of melanin. Tyrosinase is a copper-containing enzyme present in plant and animal tissues that catalyzes the production of melanin and other pigments from tyrosine by oxidation, as in the blackening of a peeled or sliced potato exposed to air. It is found inside melanosomes. It is mainly involved in biochemical reactions of melanin synthesis. Melanin is the black pigment in our skin that gives natural color to skin and hair. A mutation in the tyrosinase gene resulting in impaired tyrosinase production leads to a condition called ocular cutaneous albinism, a hereditary disorder. Tyrosinase activity is very important. If uncontrolled as in melanoma, it results in increased melanin synthesis. Several polyphenols including flavonoids or stilbenoid, substrate analogues, free radical scavengers and copper chelators have been known to inhibit tyrosinase. Molecular imprints of tyrosinase molecules contained in potentized sepia may be helped to remove the molecular errors caused by various types of inhibitors that cause certain types of albinism, leukoderma and hypopigmentations. Molecular imprints of certain chemical constituents of sepia can act homeopathically by binding to the pathogenic molecules that inhibit melanocortin receptors in melanocytes, which are the natural binding sites of melanocyte-stimulating hormones that induce production of melanin, the skin pigment of our body. Dopamine, an important constituent of sepia ink, is a neuromodulatory molecule that plays several important roles in cells. It is an organic chemical of the catecholamine and phenethylamine families. Dopamine constitutes about 80% of the catecholamine content in the brain, which is derived from its precursor chemical, L-dopa, which is synthesized in the brain and kidneys. Dopamine is also synthesized in plants and most animals. In the brain, dopamine functions as a neurotransmitter, a chemical released by neurons to send signals to other nerve cells. Neurotransmitters are synthesized in specific regions of the brain, but affect many regions systemically. The brain includes several distinct dopamine pathways, one of which plays a major role in the motivational component of reward-motivated behavior. The anticipation of most types of rewards increases the level of dopamine in the brain, and many addictive drugs increase dopamine release or block its reuptake into neurons following release. Other brain dopamine pathways are involved in motor control and in controlling the release of various hormones. These pathways and cell groups form a dopamine system which is neuromodulatory. Several important diseases of the nervous system are associated with dysfunctions of the dopamine system, and some of the key medications used to treat them work by altering the effects of dopamine. Parkinson's disease, a degenerative condition causing tremor and motor impairment, is caused by a loss of dopamine-secreting neurons in an area of the midbrain called the substantia nigra. The dopamine antagonist, also known as an anti-dopaminergic and a dopamine receptor antagonist, is a type of drug which blocks dopamine receptors by receptor antagonism. Most antipsychotics are dopamine antagonists, and as such they have found use in treating schizophrenia, bipolar disorder, and stimulant psychosis. Restless legs syndrome and attention deficit hyperactivity disorder (ADHD) are associated with decreased dopamine activity. Restless legs syndrome, also known as Willis-Ekbom disease, is generally a long-term disorder that causes a strong urge to move one's legs. There is often an unpleasant feeling in the legs that improves somewhat by moving them. This is often described as aching, tingling, or crawling in nature. Occasionally, arms may also be affected. The feelings generally happen when at rest and therefore can make it hard to sleep. Due to the disturbance in sleep, people with restless legs syndrome may have daytime sleepiness, low energy, irritability and a depressed mood. Medications used include a dopamine agonist such as pramipexole. Females are more commonly affected than males, and it becomes increasingly common with age. L-dopa, another constituent present in sepia Inc., is a precursor to the neurotransmitters dopamine, norepinephrine or noradrenaline, and epinephrine or adrenaline, which are collectively known as catecholamines. Furthermore, L-dopa itself mediates neurotrophic factor release by the brain and central nervous system. L-dopa is sold as a psychoactive in various trade names for the clinical treatment of Parkinson's disease and dopamine-responsive dystonia. Obviously, molecular imprints of L-dopa contained in potentized forms or sepia can play a big role in diseases associated with errors in dopamine and catecholamine pathways. Molecular imprints of melanin, dopamine, L-dopa, taurine, aspartic acid, glutamic acid, alanine and lysine, iodine fluorine, bromine sodium etc. contained in potentized sepia decide the diverse types its homeopathic therapeutic actions when used according to Similia similibus curantur. Taurine is a sulfur-containing amino acid that is widely distributed in animal tissues. It is a major constituent of bile and can be found in the large intestine. It has many biological roles, such as conjugation of bile acids, antioxidation, osmoregulation, membrane stabilization, and modulation of calcium signaling. It is essential for cardiovascular function, and development and function of skeletal muscle, the retina, and the central nervous system. Aspartic acid is an amino acid that is used in the biosynthesis of proteins. Aspartate has many other biochemical roles. It is a metabolite in the urea cycle and participates in gluconeogenesis. Aspartate donates one nitrogen atom in the biosynthesis of inosine, the precursor to the purine bases. In addition, aspartic acid acts as a hydrogen acceptor in a chain of ATP synthase. Dietary L-aspartic acid has been shown to act as an inhibitor of beta-glucuronidase, which serves to regulate enterohepatic circulation of bilirubin and bile acids. Glutamic acid is an amino acid that is used by almost all living beings in the biosynthesis of proteins. It is a non-essential nutrient for humans, meaning that the human body can synthesize enough for its use. It is also the most abundant excitatory neurotransmitter in the vertebrate nervous system. It serves as the precursor for the synthesis of the inhibitory gamma-aminobutyric acid. Alanine is an aliphatic amino acid, which can be manufactured by the human body, and does not need to be obtained through the diet. Alanine is found in a wide variety of foods, but is particularly concentrated in meats. Alanine has close links to metabolic pathways such as glycolysis, gluconeogenesis, and the citric acid cycle. Lysine is an amino acid that is a precursor to many proteins. Lysine plays several roles in humans, most importantly proteinogenesis, but also in the cross-linking of collagen polypeptides, uptake of essential mineral nutrients, and in the production of carnitine, which is key in fatty acid metabolism. Lysine is also often involved in histone modifications, and thus, impacts the epigenome. Due to its importance in several biological processes, a lack of lysine can lead to several disease states including defective connective tissues, impaired fatty acid metabolism, anemia, and systemic protein energy deficiency. In contrast, an overabundance of lysine, caused by ineffective catabolism, can cause severe neurological disorders. Sepia in potentized forms will naturally contain molecular imprints of dopamine, melanin, L-dopa, taurine, aspartic acid, glutamic acid, alanine, lysine etc., which fact demonstrates the role of sepia in curing multitudes of disease conditions in the living organism. A deep study of symptomatology of sepia given in various homeopathic materia medica will help us in understanding this phenomenon clearly. There are a lot of symptom groups in sepia materia medica that are similar to the symptoms of iodine, sodium, fluorine, iodine etc., which could explain by the abundant presence of those minerals in sepia ink getting absorbed from marine water environment. When potentized, sepia contains molecular imprints of all the constituent chemical molecules, which are the active principles of potentized sepia. From the above discussions, it is obvious that sepia is a highly complex drug substance that contains diverse types of chemical molecules that can play diverse kinds of important roles in the biological system. The richness of its homeopathic symptomatology also corroborates that observation. Since molecular imprints of all these constituent molecules will be present in sepia in potentized forms, it can play a very important role in homeopathic therapeutics. If you go through the mental symptoms of sepia such as indifference, aversion to family and occupation, anxiety, depression, sadness, weeping mood, irritability, lack of ability to feel natural love, always dissatisfied, sluggishness of mind, forgetfulness etc., you can see how such conditions are related with errors in dopamine pathways. This sluggishness and stasis reflects also in uterine symptoms of sepia. Persistent cold feeling in lower limbs, bruised sensation, and restlessness of legs etc. point to the condition called restless leg syndrome caused by deficiency of dopamine and errors in dopamine pathways. Skin symptoms such as chloasma, liver spots, white patches on skin, yellow saddle across nose and down the sides of face, brown spots etc. point to errors in melanin pathways. We have to explore and interpret all symptoms in the materia medica of sepia with this MIT perspective to make this study complete.